Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture we are going to talk about screening in recombinant DNA technology. In the previous video lecture, if you guys remember, we talked about the recombinant DNA technology, the overview of the recombinant DNA technology, and we talked about the vectors and some other techniques as well. Now this is also the video lecture which is regarding to recombinant DNA technology, and in this video lecture we are going to talk about screening. So let me write. Okay. Now, what is screening? Screening is a technique through which we can identify those bacterial cells which are present in a colony on agar medium plate, and such bacterial cell can contain our target DNA having the gene of interest. When we want to find out those kinds of bacteria, then through a process, then this process is called as screening. For example, we have a plate. Okay. Now inside this petri plate, what we have different kinds of colonies, like so this is the first colony, second colony, third colony, fourth and fifth. And in each colony, what we have, let's suppose in each colony there will be one colony which have all the bacterial cell that can carry the target DNA having our gene of interest. Okay, but we don't know exactly that what kind of colony having all these bacteria. Okay, we don't know. So how we know that bacterial cell? We can proceed this plate through a process that is called a screening. Through this process, the one of the colony will showing some properties. For example, again this is the petri plate, so we can proceed this petri plate through different stages. Okay, due to each stages the colony are changes, and the colony are sometimes changes. Sometimes we can load this colony into a plate which contain antibiotic, and all the colony, all the bacterial cell will be die, but some of the bacterial cell will survive. Okay, and these bacterial cell, the bacterial cell which survive, they can show that each bacterial cell can contain the recombinant DNA vector, those vector in which you have the target DNA having your gene of interest. So this is the first screening method. Okay, so second, third, and let's suppose this is another colony. Now this red color colony shows that this one colony have all those bacterial cells that can contain the recombinant DNA vector. Okay, so how we know that? We know that by a screening process. So there are several stages of the screening. So let me write each way through which we can perform the screening, and then we will explain each method of the screening in a detail in our future video lecture. So the first technique in the screening, we call that number one, colony hybridization screening. Means genetic library screening, screening of genetic library, screening of genetic library by colony hybridization. The first in of cDNA library by oligonucleotide probe. Third, we have immunological screening. Fourth, we have differential differential or plus minus screening. Fifth we have blue-white screening. Sixth we have screening by expression method. Screening by expression method. Expression. Okay. And we have some other screening technique as well, but we will explain it in our future video lecture. Okay. So in the next video lecture, we are going to talk about screening of genetic library by colony hybridization in detail. So please stay tuned and continue to watch the video. Thank you so much.